So hello students, welcome back to Vedantu NEET English, your own channel. On this channel, we do your NEET preparation in complete English. Now students, this is Basavaraj Sir, your biology master teacher. Now today is a Diwali special, one shot of cell cycle and cell division of Tejas series. Students, write down the notes. I will give you the notes on Telegram channel. Follow through and I guarantee by the end of today's lecture, you will completely master the cell cycle and cell division chapter by solving given questions. So today's lecture is going to be theory plus PYQs to make sure that whatever you're studying in my class, you will remember it until the very end. So students, if you're able to hear my voice and the beautiful presentation behind me, right, quickly fire emoji in the chat if you're able to hear me and see the presentation behind me, all of you. Now, I'll ask all of you a very simple question. Can anyone remind me, I repeat again, can anyone remind me which were the three scientists who gave the cell theory? Quickly, who were the three scientists who gave the cell theory? Okay, quickly everyone in the chat. So cell theory, first of all, let me tell you what cell theory tells you. Cell theory tells you that every single organism, animal or plant is made up of cell or a product of the cell. Okay, very good. Then came one more scientist called as Rudolf Virchow, the third one, main important one. He came and told us that every single cell arises from pre-existing cell. Yes or no? Every single cell comes into existence from a pre-existing cell. Now students, I have a pre-existing cell. From this cell, I need to make a new cell. How is that possible? What do you call the process of from one cell making a one more cell? That process is called as cell division. Clear? So all the way from the cell theory, we are learning about cell division, and that is what we are going to do in today's class, the entire chapter of cell division, where one cell will give rise to two daughter cells. Okay students? So that is called as your cell theory and the scientists involved in the cell theory and cell division. Now, all of you, can you believe that all of you at one point in your life, all of you students at one point in your life, all of you were a single cell? Can all of you believe that all of you were exactly just one cell, just one tiny puny cell? Now can anyone tell me what is that one tiny, a single cell called, which all of you were at one point in your life, starting stage of your life? That one cell is called as your zygote, is called as the zygote. Now eventually the zygote developed into, the zygote divided and it gave rise to 36 trillion cells which you are made up of right now. So from one single cell to 36 trillion cells, that is a very, very long journey. From one cell to 36 trillion cells. And all of this is possible. All of this is possible is because of a single process called as what, students? A single process called as your from one cell to 36 trillion cells. The entire process is called as your cell division. It is called as what? Cell division. So that is what is the importance of cell division. If there is no cell division, you would not probably grow. All of you will be still zygote only, high zygotes. So all of you will be still small, tiny zygote. You will not grow into a complete adult, right? So tell me, is it important to have cell division in your life? Yes, cell division is very, very important. For example, all of you are in Diwali, all of you are celebrating Diwali right now, right? All of you are playing in crackers. Now imagine, unfortunately, you get an injury while bursting some cracker. Now after some days, the injury will completely heal itself. How? A new cells are formed over the injury. How? Because of cell division. That is exactly mitosis talking, right? So if there is no cell division, all the injuries also would not heal. So the cell division plays a very, very important role in your life, all the way from the cell division to the gamete formation. Okay. So with that being said, can we start today's class, students? Are you interested now, today's class? Because the cell division is a phenomena which every single student needs to admire, not just for the sake of marks, but also to understand how the life works, how the entire life on the earth works. Okay, clear students? So this entire cell division, students, in the entire cell division, there are two main characters. The two characters of cell division. First one is called as growth. First one is called as growth. The second one is called as reproduction. Second one is called as what? Reproduction. Growth and reproduction are the twin characters of cell division. Now students, this growth and reproduction, it made me remind about a certain organism where growth and reproduction are same to same. They're not, they're same but same. Can anyone tell me which are these organisms where if it grows, it reproduces? If it reproduces, it also grows. Ring any bell? Previous chapter? Anyone? Where growth and reproduction are synonymous. It means they're same to same. From the first chapter, living world. It is mostly found in the case of your unicellular organisms. In the case of unicellular organisms, growth and reproduction are same to same. Please remember that small information which everyone needs to remember. Okay, everyone needs to remember. So before we start the class, I want everyone to know certain basics, because there are many students who are watching this lecture for the very first time. Very high chance. So if you know these concepts, I want everyone to type the answer in the chat. If you do not know, learn with me. Okay. Now what is the meaning of haploid? You might have heard about haploid. This is haploid, that is haploid, gametes are haploid. But what is the meaning of a haploid? The meaning of haploid is very simple, students. The meaning of haploid is the cell or the organism, the cell or the organism, the cell or the organism has a single set of chromosomes, has a single set of chromosomes. Very important to understand certain basic terms. For example, how do you represent, students, how do you represent haploid? You represent haploid by N. N means the number of set of chromosomes. Since there is only a single set, I will represent as N. I will represent it as N. Now what is diploid then? What is diploid? Diploid means two set of chromosomes. Example, human beings, right? So diploid is going to be two set of chromosomes. Two set of chromosomes, okay? Two set of chromosomes, and the chromosomes are going to be paired. Remember, they have paired chromosomes. Paired chromosomes. All of you know this. Paired chromosomes are going to be there. Now how do you represent diploid, students? How do you represent diploid nature? You can represent diploid nature by 2n. Very simple. 2n. Okay, 2n. Now what is chromatin? Chromatin. Remember the difference. Chromatin and chromatid and centromere, kinetochore, as well as your chromosome, everything is different. Chromatin is a thread-like structure. It is a thread-like structure. Thread-like structure. What is it made up of, students? What is chromatin made up of? Chromatin is made up of DNA plus histone and non-histone protein. Non-histone protein. Please remember all these basic terms. Non-histone proteins. Now what is chromosome, students? What is chromosome? All of you know? All of you know chromosome? Yes? Genetic structure. It is a genetic structure with condensed chromosome. It is a genetic structure with condensed DNA. Condensed DNA. This right here is a chromosome. This right here is a chromosome. Okay. Then what is a chromatid then? What is a chromatid? Here, students, in this entire chromosome, can you see this one? This one part. This single thing is called as a chromatid. This also is called as a chromatid. So there are two chromatids in a chromosome. Two chromatids. Now students, what do you mean by sister chromatids now? These two are called as what? Sister chromatid. Sister chromatid. This is the entire chromosome. These are called as spindle fibers. They are attached during the cell division. Now students, if you look around the chromosome, there is a primary constriction here. Primary constriction. The primary constriction is called as centromere. Centromere. Then we also have secondary constriction called as your satellite. Then we have telomeres, and around the centromere, we have a protein layer called as kinetochore. Kinetochore, centromere, chromatids, as well as telomere, as well as satellite. Any doubt here, students? Any doubt here? Anyone? Any doubt? Basics on point, all of you? Diwali, one shot today. Diwali one shot till now. Anyone, any doubt? If there is no doubt till now, quickly smash that like button and show your presence in the chat also. All of you. Fire an emoji in the chat. If there is no doubt till now. Now students, that was the basics which you need to understand. The next thing which you need to understand is the entire cell cycle. Ready all of you? Ready? Students, ready all of you? So what exactly happens in a cell cycle? What exactly happens in a cell cycle? There are three main events. There are three main events in the cell cycle. Okay. Students, are you liking the video? Okay. So three main events in the cell cycle is, the first one is called as cell growth. Second is called as your DNA replication. DNA replication. And the third one is called as what? The third one is called as your cell division. It's called as cell division. Okay. So these three main events in the entire cell cycle. Ready all of you? Ready? This is called as your cell cycle. This is called as your cell cycle. Now students, if you want to broadly classify, the cell cycle, I can classify the entire cell cycle broadly into two categories. Okay. Broadly into two categories. Cell cycle right here. One second students. I can divide the entire cell cycle broadly into two separate categories. Okay, done. So the entire cell cycle can be divided into two broad categories. Okay, I'll do it from here. Okay. The first one is called as interphase. First one is called as what? Interphase. Okay. Interphase. Interphase. And the second one is called as the M phase. Is called as your M phase. Okay. So first one is called as interphase and second one is called as the M phase. Now students, you need to understand here, the interphase is again further divided. Interphase is again further divided, students. Interphase is again further divided into three separate categories. Can anyone tell me in the chat what is interphase divided into? Interphase is further divided into three separate categories, that is called as G1, S and G2. G1, S and G2 are three separate categories, subcategories of interphase. Okay. Now here M phase is again further divided into two separate categories. One is called as chirokinesis. Other one is called as cytokinesis. Chirokinesis and cytokinesis. Again students, your chirokinesis is again further divided. Chiro means nucleus. The division of nucleus is further divided into multiple categories. Understand, very simple it is. Your chirokinesis is further divided into PMAT. It is further divided into PMAT. P-M-A-T. Now what is this PMAT, students? PMAT is very simple to understand. P stands for prophase. P stands for prophase. M stands for metaphase. A stands for anaphase. And T stands for telophase. Cytokinesis means, cyto means cytoplasm, kinesis means division. Division of cytoplasm is called as cytokinesis. Okay. So I'll give you a small recap of what we just learned. We learned that the entire cell cycle can be broadly classified into two separate categories, that is interphase and the M phase. Interphase is further classified into G1, S, G2. Done. Then M phase is divided into chirokinesis, cytokinesis, prophase, metaphase, anaphase, telophase, and cytokinesis as such. That is your entire classification. Now I'll give you a diagrammatic representation of how exactly it happens. Start. See here. This is your entire cell cycle. Here we have G1, S, G2. Can you see here? we have G1, S, G2, all three together. This entire structure here is technically part of interphase. Okay, interphase. Now here, do you see here this much part? This is your 𝐤𝐤kinesis, and then here we have the cytokinesis. This is the entire cell cycle. Interphase, 𝐤𝐤kinesis, where we have prophase, metaphase, anaphase, telophase, and cytokinesis. Again, one more cell cycle starts. It keeps on going in a certain pattern. Clear, all of you? Till now, any doubt, quickly ask me in the chat. I will look at the chat right now and clear any doubts possible. Quickly, everyone. Any doubt till now? Any doubt till now, students? So this is your interphase, G1, S, G2, M phase, 𝐤𝐤kinesis, cytokinesis. Very, very simple to understand. This chapter is very easy, students. So let's finish it as a butter. Okay, done? No doubt till now? No doubt till now? Amazing. Smash the like button if there is no doubt. Okay, now amazing, students. Now you need to understand this entire cell cycle takes a time duration. There's a time duration, students, and this time duration is very different in many organisms. For example, students, in the case of bacteria is different. In the case of bacteria, the entire cell cycle lasts for 20 minutes. In the case of bacteria, 20 minutes. In 20 minutes, one bacteria to two bacteria, done. 20 minutes, entire thing is over. Now in the case of yeast, 90 minutes. In the case of yeast, it is around 90 minutes. But in the case of human cell, in the case of generally the RBC, it is around 24 hours. It is going to be 24 hours. It is going to be for a time required for the cell division. Now students, I want you to understand how is this 24 hours divided. Okay. Now students, all of you are preparing for the examination right now. True. All of you are preparing for the examination. But when is the exam? Exam is in coming months. I don't want to give you all of you shock by telling reality by telling the date, but all of you know the date already. Okay. So the exam is coming near. Now, do you wait until the day of examination to study for the examination, or do you study throughout the year? All of you are studying with me right now. All of you are preparing for the examination. You're not sitting quiet in the house. Even though it's a festival, all of you are making sure that you attend the class and write down the notes and learn something new. Yes. So similarly, the cell also does not wait until the M phase. It is not simply keeping quiet. The cell also is preparing for the M phase, and the preparation stage is also called as your interphase. So out of this 24 hours, 23 hours, students, 23 hours, the cell is in interphase. It is preparing for the cell division, and just 1 hour is the actual division, that is the M phase. Is that point clear to all of you? Tell me, students, is that point clear to all of you? So out of this 24 hours, this is generally spoken as 95% of the time, and this is around 5% of the time. 1 hour is in the M phase, and 23 hours it is in interphase. Beautiful, very easy to understand. Done. That's all. We'll do concept by concept. All of you need to follow me. That's all, students. Okay. Now, students, what exactly happens in the G1, S, G2? Understand, students. Question will come from here. You cannot ignore this. So, what exactly happens in G1, S, G2 and G0 also? Understand. Okay. First one is called as your G1. Students, G1 stage. Now, how I told you, you prepare for examination. You do not keep quiet, right? So, in the G1 stage, what happens? In the case of a G1 stage, the cell is metabolically active. And it is also the first phase. Okay. Now, students, all of you remember, right? How does it go? we always start from G1, S, G2 and then M again. Then again G1, S, G2, M again. Then again G1, S, G2. Is this the process every single time? Yes. So can I tell right now your G1 stage is a stage between, M phase and S phase. Yes or no? G1 stage is sandwiched right now between M phase and S phase. True, sir? True. So technically speaking, it is a stage which is between your nuclear division, and in S phase, all of you know, in the S phase there is a DNA replication, and it is between nuclear division and DNA replication stage. Very technical this is. Very technical, but if you are able to understand, awesome it is, awesome. So it is a stage between nuclear division and DNA replication stage. Very beautiful line, very beautiful line. The cell is metabolically active. The cell is active. The cell is active here. The cell is metabolically active. Okay. The cell is metabolically active. The cell is metabolically active here. But students, is there DNA replication here? No. No DNA replication. There is completely no DNA replication here. No DNA replication. No DNA replication. There is completely no DNA replication here at all. Okay, that is G1. Now, what exactly happens in the case of S phase now? S phase. In the case of S phase, what happens? First main point, everyone listen to me very carefully. It is the second stage. Can I tell S phase is a stage which is between G1 and G2 stage? Yes, it is between G1 and G2 stage. Okay. Now, what is the main process here? The main process here is DNA replication. DNA replication. Now, what do I mean by DNA replication? The meaning of DNA replication is that increase in DNA content. Increase in DNA content. There is an increase in DNA content here. Now, what do I mean by increase in DNA content? All of you focus here. All of you focus. Extreme focus now. All of you focus now. Here in the G1, this was the DNA in the single chromatid. Okay, single chromatid. For example, let's say this is 2C. 2C is the DNA content. 2C is the DNA content here. Now, as soon as we come to the S phase, what happens? As soon as you come to the S phase, what is happening here? As soon as you come to the S phase, the DNA content is doubling now. The DNA content is doubling. Can you see here? This is the from 2C it becomes 4C. 2C to 4C, increase in DNA content. Increase in DNA content. So, why is there no DNA replication G1 stage? Because G1 stage do not have the required proteins or required mechanism for the replication. G1 stage is a very simple stage where the cell is growing. Simple. Because you can, before you grow, you need to prepare. For example, if you want to go to a trip, do you pack first or do you randomly go? You pack, right? You do some packaging, you prepare for the trip. In G1 stage, the cell is preparing. Preparatory stage. S stage is where the cell division happens. Now, there is a very famous question here. All of you need to listen to me very carefully. The famous question here is there a change in the number of chromosomes? See students, listen to me very carefully. There is DNA content is increasing. There is always DNA content is increasing, but the DNA or the chromosome number is not changing. Chromosome number is not changing. There is no change in chromosome number. No change in chromosome number. No change in chromosome number. So, if the chromosome number is going to remain as 46 only, the number of chromosomes are going to remain the same itself. Okay. So, chromosome number is same. The chromosome number is same. No change in chromosome number. Very important point. Many students make the mistake here. So, if it is 2n here, if it is 2n here, so what will happen here, students? Tell me, students. If it is 2n here, here also we will have 2n itself. There is no change in chromosome number. It is going to remain as 2n itself. 2n to 2n. Clear? Very important to understand. Very, very detailed concept it is. Small errors can make you go wrong here. Okay. So no change in chromosome number. The last point here is the duplication of centrioles. Duplication of centrioles in cytoplasm, in the cytoplasm. Now students, tell me, is this duplication of centrioles, does it happen only in plants or only in animals, or does it happen in both plants and animals both? Tell me, students. If it is called as duplication of centrioles here, does it happen only in the plants, only in the case of animals, or it happens in both animals and plants? Quickly answer. Students, remember, the centriole is a structure which is found only in the case of animals, only in the case of animals, not in the case of plants, not in the case of plants. Okay, so G1S, G2 is very simple, students, nothing much happens here. In the case of G2, the cell grows. I'll write here on the top because G2 nothing very special. G2, the cell grows. The cell generally grows, and also the only point where the question can be asked is production or synthesis of protein. Synthesis of protein can take place here. Proteins. Mostly proteins happens here. Protein in a certain RNA. Okay. So till now, any doubt, students? Any doubt till now? G1S, G2, done. That is between nuclear division and DNA replication stage. The cell is metabolically active. No DNA replication. Between G1 and G2 stage, DNA replication, increase in DNA content, no change in chromosome number, replication of centrioles in the cytoplasm. Happening in nucleus. These are happening in nucleus, DNA replication in nucleus. Then G2 is cell growth, production of synthesis of protein. Okay. So that was your cell cycle interphase. Now students, there is one more stage called as G0 stage. See here. Now students, in your I'll tell you one sad story. Once you give your, just like how you cannot give your heart to everyone else in the world, only to a very special person, the heart cells are also very special, because these heart cells do not divide very well, do not divide. Heart cells are a little exception to the cell cycle. Okay. Heart cells are such an exception that they exit the cell cycle, or they do not undergo the entire cell cycle. They do not divide at all, heart cells. Okay. So what exactly happens here? So if you look here, after G1 here, can you see here? After G1, some cells come out of the G1 stage. They exit after G1 stage. They exit after G1 stage, coming out, and that is called as G0 stage. That is called as G0 stage or quiescent stage. The quiescent stage or G0 stage. Now students, you need to understand here, even though the cells are going to be inactive, but they are going to every single time there is a requirement, they can become metabolically active and they can come back, and they can come back to you. Okay. So every single time there is a requirement, the cells can be called upon for. Okay. Clear till now, students? Any doubt till now? Any doubt till now, students? So let's see what we finished cover till now. I'll give you a small recap. We finished about the entire cell cycle. We learned about cell cycle, what exactly happens here. That is DNA replication, cell division and cell growth. That is your cell cycle. Cell division, DNA replication and cell growth. Okay. Then we learned about interphase. 90 minutes for the cell cycle to takes place in the case of yeast, 24 hours in the case of your human cell. Yes, we did that. Then we did something called as your chirokinesis and cytokinesis division. G1S, G2. In G1 phase, what happens? The cell is metabolically active, okay, but does not replicate DNA. S phase or synthesis phase. Friends, G1 is called as gap 1. S is called as synthesis.
Okay, synthesis phase. Okay, see 2C2, 4C, the DNA content is doubling. Okay, in animal cell during the S phase, DNA replication begins in the nucleus, the centrioles duplicate in the cytoplasm. In animal cell, not in the case of plant cell. Okay, now students, then we learned about G0 stage. Okay, see heart cell that do not appear exhibit division. Heart cell cannot divide. Okay, do not further they enter G0 stage. Okay, see the cells in this stage remain metabolically active. They are metabolically active. Okay, but no longer proliferate. It means that they can divide, but they chose not to divide. You can study, but you decide to be lazy. You can study, you can score really well, but due to some laziness, you do not want to do so. Okay, there is no requirement for you. Okay, so here also, the cell remains metabolically active, but no longer proliferate unless called upon to do so. Okay, done students? Done. Now, let's go to the main show now. Can we start? The main show called as mitosis. Can we start students now, mitosis? Till here any doubt? Very beautifully covered, beautifully covered. Now, let's finish the entire mitosis in 15 minutes. Entire mitosis in 15 minutes. Can I get ultimate attention for the next 15 minutes? Entire mitosis you will learn and you will go home, be happy later on. Then we'll do meiosis today only. One shot today, one shot. Okay. So, the next 15 minutes I want everyone to pay extra attention. Every single time I ask a question, reply to me, you will get a better idea. Okay. Now students, the entire mitosis is divided into two categories. I repeat again, the entire mitosis is divided into two separate categories. One is called as 𝐤𝐤kinesis, other one is called as cytokinesis. So what exactly happens in 𝐤𝐤kinesis? 𝐤𝐤kinesis is further divided into. 𝐤𝐤kinesis is further divided into prophase, metaphase, anaphase, telophase. That is PMAT, P-M-A-T, prophase. Repeat after me, prophase, metaphase, metaphase, anaphase. All of you repeat after me, anaphase and telophase. These are the four different, 4 different stages in the 𝐤𝐤kinesis. 4 different stages in the 𝐤𝐤kinesis. PMAT, PMAT, done. PMAT. Now what exactly happens in the case of prophase? Prophase is the first stage in the cell cycle, first point. It is the longest phase in the cell cycle, second point. Okay. Now what exactly happens in the prophase? Now students, you need to understand, in the interphase, the DNA was present in the form of chromatin. I repeat again, in the interphase, DNA was present in the form of chromatin. Now this chromatin, the thread-like structure, this chromatin, which is a thread-like structure, undergoes something called as condensation. So the chromosomes starts to condense to form the chromatin, start to condense to form the chromosome. That is initiation of condensation of chromosome material. So what exactly is happening here? In the case of prophase, the chromatin, which is a thread-like structure mostly, okay, it starts to condense and it results in the form of a chromosome. Okay, so here it is divided into early prophase and late prophase. In early prophase, what is happening? Condensation of chromosomes, first point. Movement of centrosome. Students, what is centrosome made up of? Tell me in the chat, students. What is centrosome made up of? Centrosome is made up of centrioles. So movement of centrosome. Can you see? The 2 centrosomes are here. How many are, students? 2 centrosomes are here. Now you tell me, if there are 2 centrosomes, how many centrioles do you find in one cell? Tell me. In one division, how many centrioles do you find? 2 here, 2 here. So totally 4 centrioles. You find here. Okay. Now students, you look at the diagram of centrosome here. In the centrosome, there are 2 types of microtubules. Can you see here? There are 2 types of microtubules. The first one is called as asters. Can you see here? The pink color one is called as asters. We call them asters. We call this one as aster formation, aster. The one which is present on the top, the blue color one, this is called as spindle fiber. It's called as spindle fiber. Okay, spindle fibers. So there are two types of microtubules, asters on the top and spindle fibers. Asters mainly help in anchorage. Spindle fiber attached to the entire chromosomes. Asters are found only in the case of animals. Spindle fiber can be found in both in animals and plants. Very important point. Notes point to point. Spindle fiber present in both animals and plants. Asters only in the case of animals. Now students, you need to understand the next point is asters plus spindle fiber together they are called as what? Mitotic apparatus. They are called as what? Mitotic apparatus. Together they are called as mitotic apparatus. Now students, this is called as your early prophase. First point is what? Condensation of chromosomes. Second point was movement of centrosome. Centrosome is made up of asters and spindle fiber. Together they are called as what? Together they are called as mitotic apparatus. Okay. Now late prophase, what is happening? Nuclear membrane is disappearing, Golgi complex disappearing, endoplasmic reticulum is disappearing, everything is disappearing, preparing for the cell division. Prophase clear or not? Tell me in the chat, all of you. Prophase clear or not? Tell me. All of you done prophase? Done? Okay. So this is your entire prophase. Condensation of chromosomes, movement of centrosome, done. Now students, next thing you need to understand here is going to be diagram given in NCERT. See, early prophase, late prophase. Can you see in the late prophase we have the entire chromosomes. Entire chromosome is there. Nuclear membrane is disappearing. Nuclear membrane is disappearing, late prophase. Then we have metaphase. What exactly happens in the case of metaphase? In the case of metaphase, all the chromosomes are coming and aligning in the middle. Can you see? All the chromosomes are aligning in the center. Aligning in the center. The first point is what? First point is movement of chromosome. Movement of chromosome. Formation of metaphasic plate, also called as equatorial plate. Can you see? The middle point is called as what? Metaphasic plate or equatorial plate. Here we have spindle fiber attachment. Now students, the main point here. The main point here. This is the chromosome. Primary constriction called as centromere. Around the centromere we have the kinetochore. This is called as kinetochore. Now students, all the spindle fiber, do you see here? All the spindle fiber which is attaching from here, 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 there will be spindle fibers going around also. Yes or no? So the spindle fibers will attach to the chromosome. Spindle fiber will attach to the chromosome. The point of attachment is called as kinetochore. So students, see this. All the spindle fiber will attach to the kinetochore. Will attach to the kinetochore, but where? Kinetochore in the metaphase. In the metaphase. So attachment of spindle fiber to the chromosome happening at the kinetochore. Is that point clear to all of you or not? Tell me yes or no. Yes or no. Sir, compaction of mitotic chromosome means compaction. Compaction means condensation. Condensation of chromosome. Okay. Done. Now students, you need to remember this as this entire structure, this entire process is called as mitosis. And this mitosis mainly happens in the case of somatic cells. The normal cells in the body, skin cells, muscle cell, cheek cell, somatic cells. Okay. Metaphase done, all of you? Yes. See, spindle fiber are attached to the kinetochore. Next point is best stage to study this. Study chromosome morphology. To see the chromosome morphology, best stage. Best stage. Done students? Done? Done? Amazing. So in the case of metaphase, all the chromosomes are aligning at the equatorial plate. Done. Now what exactly happens? See, this is the transition to the metaphase. Metaphase. All the chromosomes are aligned at the metaphase. Spindle fiber attaching where? Spindle fiber attaching to the kinetochore. To the kinetochore. To the kinetochore. Done. Now students, this is the most important part of the mitosis. The most important. That is called as your anaphase. So what exactly happens in the case of anaphase? Understand very carefully. Here we have the chromosome. Here we have the chromosome, yes or no? Chromosome. Now students, I want you to imagine, I am one chromosome here. One spindle fiber is attaching from here. One spindle fiber is attaching from here. Now imagine from here 10 people are pulling, from here 10 people are pulling. So both people are pulling my hands. What will happen? Will I split into half? Yes or no? So people if you start pulling from here, eventually will I split open? Yes. So same thing is happening here. Imagine here I have the kinetochore. Around the kinetochore I have the spindle fiber. Now if I start pulling from each side, what will happen? The centromere, the middle part, the centromere will split, will completely split. When it completely splits open, can I get two separate chromatids now? Yes or no? Tell me. Yes or no? Obviously. Right? So if I split open, two chromosomes separate, two chromosomes separate. That's all. That is your anaphase. Now till here it is very easy to understand. What is happening here. In the case of anaphase, sister chromatids are separating. In the case of anaphase, shortest phase, sister chromatids, they are called as sister chromatids, are separating. Is that point clear to all of you? Tell me. Is that point clear to all of you? In the case of anaphase, separation of sister chromatids. So if I ask you a question, all of you question, in which phase of mitosis, separation of sister chromatids takes place? What should be your answer? Students? Answer should be anaphase. Now if I ask you, in which phase of your mitosis chromosomes align at the equatorial plate, then what should be your answer? Answer should be metaphase. In the case of metaphase, all the chromosomes are aligning at the equatorial plate. Now if I ask you one more question, in which phase of cell cycle, that is in mitosis, spindle fiber are attaching to the kinetochore, answer is metaphase again. Okay. Now here I want you to understand one small thing. Can you see here? Chromosomes are being split. One sister chromatid this side, one sister chromatid the other side. Separation of sister chromatids is happening. Now students, you need to understand here is one important point. That is, they are called as daughter chromatids. Now eventually, tell me, this will split into half. This will become one cell, this will become one more cell eventually. Yes sir. One cell, separate cell will become. Yes. Now students, eventually can I tell this entire chromatids will become chromosomes? Yes. So the daughter chromosomes, can I tell this daughter chromatids will eventually become daughter chromosomes in the future nuclei. They will become daughter chromosomes. So technically speaking, right now in anaphase, every single chromatid is acting like a chromosome. I will repeat again. The chromatid will become chromosome eventually. So at this point, temporarily, shortest phase, what is happening? Every single chromatid is acting like a chromosome. Very important point. Chromatid acting like a chromosome. That is, in total of how many chromosomes do we have, students, here? In total we have 46 chromosomes. Every chromosome has 2 chromatids. 46 into 2 is how much? 46 into 2 is 92 chromatids. In total we have 92 chromatids. 46 chromosomes into 2, 92 chromatids. Here, this is one chromosome. Every chromosome has 1, 2 chromatids. So totally 92 chromatids. So here we have 92 chromatids. Every single chromatid will act like a chromosome. So technically speaking, temporarily at anaphase, how many chromosomes do we have? Tell me, students. Temporarily at anaphase, how many chromosomes do we have temporarily? 92 chromatids equals 92 chromosomes. So temporarily at anaphase we have 92 chromosomes temporarily. Beautiful. Now best stage to study the chromosome structure, that is metaphase, metacentric, submetacentric, telocentric, acrocentric, all that can be studied best at the anaphase, Beautiful. Last stage is called as your telophase. Okay, last phase is called as telophase. So what exactly happening in the case of telophase? In the case of telophase, we have chromosomes cluster at the poles. All the chromosomes are being clustered at the poles now. Identity is lost because they are again turning back to chromatid structure because cell division is over now. Okay, nuclear membrane reappears, Golgi complex, ER, everything is reappearing, and the entire nuclear division is over. So at the end of telophase, what is happening? All the chromosomes are clustering, and nuclear membrane, Golgi complex, everything is again reappearing. And that is the beauty of mitosis. See here, telophase. Okay, now next thing is called as cytokinesis. Obviously, if nucleus is divided, we should also divide cytoplasm. Yes or no? Tell me. If you are dividing the nucleus, cytoplasm also needs to be divided. Yes or no? So cytokinesis is called as division of cytoplasm. Okay. Now students, this division of cytoplasm is very different in the case of your animal cell and plant cell. Very different. Now students, how many of you played with balloons? Tell me, raise your hands. How many of you like playing with the balloons? Bursting balloons, throwing on each other. Tell me. How many of you like bursting balloons? Okay. Now students, I'll give you. Okay. Now see here, students. If I take one round balloon, if I start pressing the two balloons from the other side, both the sides, at one point, if it is a very large balloon or if it is a balloon which is not very completely filled with gas or air, at one point the two balloons will meet. Yes, they'll meet. Or the clay. All of you know clay, right? Now if you take a clay ball and you try to pinch the clay ball from the two sides, eventually it will meet. Now that is what is happening in the case of your animal cell. In the case of animal cell, we have something called as microfilaments. Okay. Now here chirokinesis is over. Now what do? You pinch from both the sides. You're pinching from both the sides. Now this is called as what? This is called as furrow formation. Can you see here? Can you see this gap here? This is called as furrow formation. Now eventually you will keep on pinching it, and eventually if you keep on pinching it, you will get two different cells. Yes, eventually that is called as centripetal manner of cell division or cytoplasm division. When you pinch from the two sides, furrow formation happens and you get two cells. Now you cannot do the same thing for your plant cell. Can anyone tell me why? You cannot pinch the cell wall? You cannot pinch the plant cell. Why? Why, students? Why you cannot pinch the plant cell? Very easy, students, very easy to understand. For example, imagine this is a brick. Right here. Now, can I pinch it? It's very difficult to pinch, right? It's very difficult to pinch this brick. Very difficult. How much ever you try from outside, the pinching will not happen. Why? Because around the cell, there is a very rigid wall. What is that rigid wall called as? The rigid wall is called as your cell wall. It is called as cell wall. And the same cell wall is absent in the case of animal cell. So very easy work was there. Very easy work. But in the case of your plant cell, there is something called a cell wall, and it is very difficult from the outside. Now students, in your house, okay, during festival, mother will scold you first, okay, outside they'll hit you one shot on the head shot one, okay, and you will not care. Mother will, mom will scold you nicely from outside, you're like, scolding will happen every single day. Now tell me, what is the ultimate weapon of every single mother apart from chappal? Tell me, what is that ultimate weapon of your mother other than the chappal? Tell me, can I call it emotional blackmail? I gave birth to you. I gave birth to you. You cannot do this much for me. That emotional one blackmail will come. Yes or no? The emotional blackmail happens from the inside. Yes, from inside it happens. Same thing we are doing to the plant cell. Since plant cell, you cannot go from outside. How much ever you press from outside? Not bulging. Why? Because of the cell wall. Because of cell wall. Not happening. Now what do? From the inside you again now you go. Now what do? Inside there is something called as cell plate formation. Can you see? On the inside we have the cell plate formation. Cell plate formation on the inside. Can you see? Cell plate formation on the inside. Now you tell me students, if I make the cell plate on the inside, if I make a cell plate on the inside, now can I divide this way and this way? The cell plate will eventually divide the two cells. This will move this side, this will move this side. Eventually we will get two cells. Eventually we will get the two cells. Beautiful. That is called as cell plate formation in the case of plants. That is centrifugal manner. It is called as what? Centrifugal manner of cell division from the inside to outside. Beautiful, beautiful. So that is the end of your cytokinesis. Now students, I'll ask all of you one simple question. How many of you want to learn the DNA content? Raise your hands. How many of you want to read the DNA content? Learn the DNA content. It's a very technical thing to understand, but if you understand, you will never go wrong in the cell cycle. Never go wrong in the mitosis. Can we start DNA content? Do you want to learn DNA content, students? Tell me. Raise your hands. DNA content. Now students, now I will prove you. Now I will prove why do you call mitosis as equational division. Can we start? Why do we call DNA mitosis as equational division? Can we start? Okay. Tell me students, the entire cell cycle starts with what? The entire cell cycle starts with G1. G1. Okay. Here we have our chromosome. Single chromosome in the case of G1, and I will call it 2 and 2C. 2 and 2C. 2 and 2C. Okay. Now then we have S. What exactly happens in the case of S? We have DNA replication. We have DNA replication. So 2 and 2C, 2 and 2C will become 2 and 4C. Change in DNA content? Yes sir, change in DNA content. Then we have G2. G2 will remain the same. G2 will remain the same. 2 and 4C. In fact, in the case of prophase, metaphase also it will remain the same. It will remain the same. But as soon as we come to anaphase, what happens? As soon as we come to anaphase, one chromosome, tell me, in the anaphase, is it not splitting into half? Yes or no, tell me. It is splitting into half. It is splitting into half. So what do we have here? We have one chromatid and one more chromatid here. Now did we learn that? Each single chromatid is acting like a chromosome here. This is a chromosome. This is a chromosome right here. Okay. This is a chromosome right here. Okay. This is a chromosome right here. Now, students, understand this very simple concept. Here we have 2N. Why do we write 2N? We write it 2N because there is 46 chromosomes. 46 chromosomes. 46 chromosomes. Okay. 2n, 2n, 2n. But in the case of anaphase, how many chromosomes did we have, students? In the case of anaphase, how many chromosomes did we have? Can we go back and check? In the case of anaphase, did we have 92 chromosomes? Because 92 chromatids were acting like chromosomes. Yes or no? So if 2n is 46, if 2n is 46, what is 92? Tell me. If 2n is 46, tell me, what do you call for 92? What do you call for 92, students? Can I call 92 as 4n now? 4n, obviously. So right now at anaphase we have 4n, 4c. Okay, 4n, 4c. Done. Then we have telophase. Then we have telophase. So in the case of telophase, what is happening? In the case of telophase, the cell is dividing into two, right? Here we have, out of 92 chromosomes, out of 92 chromatids, can I tell 46 went this side and 46 went this side? Out of 92 chromatids in the case of anaphase, 92 went this side, 46 went this side and 46 went this side. Yes, sir. So can I tell this 2 and 2c, got divided into 4 and 4c got divided into 2 and 2c this side and 2 and 2c this side. Yes or no? Tell me. Yes or no? So 4 and 4c became 1, 2 went this side, 1, 2 went this side. From 4c, 2c went this side, 2c came this side. Yes or no? Yes or no, all of you? So if you take the entire cell right now, because in telophase it is still dividing, right? If you take the entire cell, here, the cell is still dividing, right? If you look at the entire cell, it is still 4 and 4c. But if you look per nuclei, it is 2 and 2c. This is the entire nucleus. One side is 2 and 2c, other side is 2 and 2c. Now students, you tell me students, we started off with 2 and 2c, we are ending with 2 and 2c, starting with 2 and 2c, ending with 2 and 2c. From one cell, two cells. From one cell which is 2n, we made two different cells which both are also 2n. 2n, 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 2n. So can I call it equational division? Equal number of chromosomes from the parent to the offspring, no change in chromosome number, but there is a division in the cell, that is called as equational division. That's why we call mitosis as what? Mitosis is called as equational division. Equational division. Done? Beautiful. All of you very smart nowadays. Okay. Friends, here is something called as significance. I will write few points. They'll ask you directly question here. So write down these points. All of you are smart students now. You are learning from me this concept. You cannot go wrong after this. Okay. Now before we read, mitosis or equational division is usually restricted to the diploid cells only. Now what is the meaning of that? Mitosis will happen only in the case of diploid cells, generally diploid cells, 2 and 2n. Okay. However, in some lower plants and in some social insects like haploid cells also divide by mitosis. Okay. So in the case of lower plants, the meaning of lower plants is algae. In the case of algae, even the haploid cell undergo mitosis. Haploid cell also undergoes mitosis. Also seen in the case of drone, male honeybee. In the case of drone also undergoes mitosis to produce the gametes. Okay. See, now mitosis usually results in the production. of the diploid daughter cells with identical genetic complement, meaning there is no recombination. The growth of multicellular organisms is due to mitosis, the first point right here. Okay. cell growth results in the distribution of ratio between the nucleus and cytoplasm. It is therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. Very important point, very important point. Restoration of nucleocytoplasmic ratio. Okay, restoration. Now what do I mean by this, students? Understand. What is the meaning of restoration? Understand. Here I have one cell. For example, this is the cell. Okay. Now here I have a large nucleus. This is the cytoplasm. Now imagine the cell is growing. Imagine the cell is growing here. Okay. The cell is growing. And this is the cytoplasm now. This is the cytoplasm. And this is the nucleus. So tell me, students, here the ratio is maintained. But as soon as the cytoplasm became big, the nucleus is very tiny. Yes or no? Very tiny. Now what do? To maintain the nuclear and cytoplasmic ratio, you divide the cell. So what happens eventually? You get one cell here. And in the middle we have the nucleus again. Nucleus. So the balance between the nucleus and the cytoplasm, the ratio is maintained. The ratio is always maintained. Okay. Beautiful. The ratio is always maintained. The nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. Very important, cell repair. Third one. The cells of the upper layer of the epidermis, cells of the inner lining of the gut, the blood cells are being constantly replaced. Okay. Fourth point is meristematic tissue. We tell, right, plants can keep growing, indeterminate growth. That is because of meristematic tissue, cell division. The apical and lateral cambium results in the continuous growth because of plants through the lifetime. So four main significance of mitosis. Done. Tell me the answer to this question, students. Tell me the answer to this question. Let's see how many of you can tell me. Very simple question. Let's see how many of you can tell me. Time starts now. Students, quickly smash that like button, all of you. And students, if you want these notes, all the notes will be available on the Telegram channel. Let me tell you now only. All the notes will be available on the Telegram channel. If you want the notes. You don't want notes, good for you. The spindle fiber attached to kinetic core of centromere during metaphase. Very simple question, very easy answer. The following are the stages. See, students, this is a NEET 2024 question. I am not giving you deliberately easy questions. Remember, all of these questions are NEET PYQs. The following are the stages of cell division. What do you have first? First we have G1, S, G2, Ch, Cyto. So E will come first. Okay, this is gone. So EC, right? EC. 3 is the answer. Okay. Next question, see this fast. What exactly happens in G1, S, G2 and Z0? Okay, G1 metabolically active, grows in size, synthesis of protein, happens in G2. Replication of DNA, S. What is the answer now? Tell me. A is going to be 4, 2. Next question. Tell me, students. Tell me quickly. Among eukaryotes, replication of DNA takes place in, very easy question, S phase. Next question. M phase, what happens, students? M phase is called as equational division. A is 4. A is 4. These two are gone. G2 phase, what happens? Synthesis of protein. Okay. So answer is 4. P is 1. C is 2, obviously. Then C is going to be 2. Quiescent phase or G0 phase. G1 is what? Interval between mitosis, that is the M, and initiation of DNA replication, that is S phase. Done. Next question. Students, quickly smash the like button. During G0 phase of cell cycle, the cell is metabolically inactive. Metabolically inactive, students. Tell me, is it proper? It's going to be active. The centrosomes undergo duplication in the S phase. Yes. First is incorrect. Second is correct. See, it is metabolically always active. Metabolically active. Next question. Which of the following is a quiescent stage, that is G0? Quiescent stage is G0. Which of the following never occur during mitotic cell division? Mitotic cell division. Coiling of fertilization of chromosomes happens prophase, metaphase. Movement of centrioles from the opposite pole, prophase. Pairing of hormone does not happen. This does not happen because there is no homologous chromosomes. The concept of homologous chromosomes only happens in the case of your meiosis. Meiosis. Okay. Select the incorrect statement with refers to mitosis. Okay. Splitting of centromere occurs at anaphase. Yes. All chromosomes lie at the equatorial plate in metaphase. Chromosomes lie. Okay. Spindle fibers attached to the centromere. No. It attached to the kinetochore. Not centromere, kinetochore. Yes. D contains the telophase. Done. 2022 question. Next question. With respect to metaphase, the following statement is incorrect. Incorrect. Okay. Complete disintegration of nuclear membrane has taken place. Yes. Chromosomes are highly condensed. Yes. Metaphase chromosomes are made up of forced chromatids. No. 2. 2. 3 is the answer. Easy question. The centrioles undergo duplication in. The centrioles undergo in the cytoplasm. In the cytoplasm, they undergo duplication and in the S phase. In the S phase. During which phase of the mitosis do the chromatid becomes chromosome? I told you. Each individual chromatid will act like a chromosome. Will act like a chromosome. Which stage, tell me? It will act like a chromosome. In the anaphase. Beautiful. Next question. Some cells enter G0 phase leading to the inactivation of the cell cycle. Correct. G0 phase occurs due to non-availability of mitogens and energy-needed compounds. Yes. Mitogens are the chemicals which help in mitosis. Okay, that's why it happens mainly. Correct. Okay, students. So that is the end of mitosis. Any doubt in mitosis, students? Let me see. Let me see. Anyone? Any doubt in mitosis? Quickly. I'm looking at the chat right now. Looking at the chat right now, students. Any doubt in mitosis? Quickly smash that like button first of all. Okay. And any doubt in mitosis, students? Any doubt in mitosis? Any doubt in mitosis? Done. If there is no doubt in mitosis, can we start meiosis now? Next 1 hour, meiosis will finish it off with questions. Okay. Can we start? Can we start? Yes. Beautiful. Now students, next we will go to the meiosis now. Very simple meiosis. The first thing you need to understand about meiosis is where does it happen? Where does it happen and why is it important? Okay. Students, remember, meiosis is a phenomena which mainly happens in the case of germ cells. It happens mainly in the case of all the germ cells. Okay, one second students, let me check the chat. It mainly happens in the case of germ cell. Can anyone give me an example of germ cell? A quick example of germ cell. It could be spermatogonia, oogonia. Spermatogonia, oogonia undergoes meiosis to produce the gametes. It undergoes meiosis to produce the gametes. Whereas the germ cell is said to be diploid. The gametes or the result of meiosis is always going to be haploid. 2 into 1. Now tell me students, is there a change in the number of chromosomes here? Yes sir, there is a change in the number of chromosomes. Chromosomes are decreasing here. Change in the number of chromosomes. And one more thing you need to understand here is, from one diploid cell, we will be able to obtain 4 haploid cells. That is N, 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 N. So from one diploid cell, we will be able to obtain 4 haploid gametes here. 4 haploid gametes. Clear? Now, what is the significance of your meiosis? Students, understand, if there was no meiosis, what would happen? Small idea, imagine. Here we have germ cell which is diploid. For example, this germ cell is from male. From female also we have one germ cell. Okay. If there was no meiosis and only mitosis, gametes will be 2n. Gametes will be 2n. Gametes will fuse, we will get 4n. The organism will be 4n now. This 4n organism will also produce gametes, that also will be 4n. Here also we have 4n, for example, 4n. Now offspring will be 8n again, 8n. So do you realize, if there was no meiosis, then there would be no proper, the ratio would not be maintained and will become polyploidy. Will become polyploidy eventually, and there would be a very big chaos. So checkpoints, Ravindrani, checkpoint is very simple. Do you want to learn about checkpoints, students? Do you want to learn about checkpoints? Let me teach you today. Usually I don't teach checkpoints, but today I am feeling the little extra happy because of festival season. Do you want to learn about checkpoints, students? Tell me. Do you want to learn about checkpoints? Okay, I'll teach you today checkpoints. It's very simple. There are 2 main checkpoints, students. Understand? Oops. 1 second. The mic only came off. So today is Diwali. So I will teach some extra points today. Understand? In the case of a cell cycle, there are many factors there. What you learn right now in the cell cycle which you are learning in your class 11th level, class 12th level, this is the most basic form of cell cycle. Don't be like, this is the only cell cycle which you know. No. Cell cycle is a much more very complex process, and there are so many signaling involved, CDK cycles are involved, many proteins are involved, everything is involved. What I teach you right now for your benefit is the most basic form of cell cycle. Okay. Now today I will teach you one small extra concept called as checkpoints. Okay. Now students, in examination, when you are writing a paper, after finishing one entire, for example, you are writing your NEET paper, after finishing the bio part, will you again cross check your answers? Tell me, will you cross check your answers again? Yes sir, you will cross check your answers. Here also, in the entire cell cycle, there are certain checkpoints to make sure the entire process is very smooth. Okay. For example, the first checkpoint is after G1. The first checkpoint is after G1 called as G1S checkpoint. G1S checkpoint right here. Now, what is exactly happened this checkpoint here? At this checkpoint right here, there is a checking. What is checking happening here? The checking is happening, that is the sufficient nutrients are there for DNA replication? Is the cell ready for DNA replication? Do we have enough of proteins for DNA replication? So basically, the first checkpoint makes sure to make sure that the DNA replication happens very smoothly. Every single requirement is there, because all of you know, right, in DNA replication we need helicase, we need topoisomerase, we need single stranded binding protein, we need DNA ligase, we need DNA polymerase 1, we need DNA nucleotides, DNTPs. So in the first checkpoint we are making sure all of these are already there. First checkpoint. Okay, so first checkpoint is done. In the first checkpoint we are making sure everything is done. So DNA replication takes place here. DNA replication. And those of you want to know what is topoisomerase, what is DNA helicase, what is DNA ligase, what is SSB protein, and what is your what is the meaning of Tata box, what is the meaning of what is that, DNA polymerase 1, what is the energy, where is the energy coming from, all this you will study in the molecular base of inheritance chapter, not here. Molecular base of inheritance. Okay, in inheritance. Now what is happening in the second checkpoint? The second checkpoint is between what? The second checkpoint is called as G2S checkpoint. G2S checkpoint. Here we have G1S checkpoint, or we can also call this SG2 checkpoint. Okay. Now this checkpoint makes sure that DNA replication is happening very smoothly. DNA replication is happening very smoothly. Now what if there is a error in DNA replication, students? What will happen? Tell me. What if there is a error in DNA replications? Cancer cells have to check. Okay, tell me, what will happen if there is no checkpoint after S? What if there is an error in DNA replication? That could lead to mutation. Tell me, that could lead to mutation. So in the case of cancer cells, what happens? The checkpoints are affected, right? Uncontrollable growth in the cancer cells. Okay. So in the second checkpoint, we are making sure. In second checkpoint, we are making sure that the entire DNA replication is going very smoothly, and if there are no errors, before we go to the next concept, before we go to the next phase, making sure that all the replication is proper. Now, can anyone tell me, if you have studied the molecular base of chapter, inheritance chapter, if there is an error in DNA replication in your prokaryotes, who is correcting it and how do we correct it? Tell me, molecular basis, you have studied molecular basis, no? I'm checking your knowledge now. From that chapter we learned that your DNA polymerase 1, DNA polymerase 3 itself, DNA polymerase itself completely scans. DNA polymerase itself scans the entire DNA strand, and every single time there is a mistake, your restriction endonucleases, with the help of restriction, not endo, restriction nucleases, we will come and we will cut the restriction endonucleases, will cut it and we will remove the extra part and throw it out. Okay. So remember that point, The second checkpoint is called as SG2. Happy? All of you happy for Diwali now? And students, let me know in the chat if you want to learn. Let me know in the chat if you want to learn the molecular base of inheritance chapter someday. Okay, I will teach you that also. It's very interesting chapter, students. Very interesting chapter. Okay, very interesting chapter. Now, students, let's start with meiosis now. We started, but we went a little deviation. But let's start with meiosis right now. Meiosis. Now, all of you know the basis of meiosis now. All of you know basics of meiosis right now. Basics of meiosis. Very basics of meiosis you know. That is, germ cell, gametes, meiosis takes place. Now, students, if I want to broadly classify meiosis, the entire meiosis can be divided into two separate categories. Okay. So meiosis, let's get it. Meiosis. Okay. Meiosis. The first category is called as your interphase. And the second category is called as meiosis. Okay, meiosis. Now students, interphase, what do we have students in the interphase? In the interphase we have G1 S G2. We have G1 S G2. Okay, G1 S G2 we have done. In meiosis, you can students, the further meiosis is again further divided into three separate categories. Three separate categories. First we have meiosis 1, then we have meiosis 2, and in the middle we have interkinesis. Now this meiosis 1 also is further divided into PMAT, that is PMAT is going to be prophase, metaphase, anaphase and telophase, and all this is 1 and 1. Prophase 1, metaphase 1, anaphase 1, telophase 1. Done. Now here also we have what? PMAT. Everything is going to be 2 here. Okay. Now, what about interkinesis? Interkinesis is just like your interphase, very similar to interphase, G1 is G2. Okay. But there is a one small difference in interkinesis. Interkinesis is very short-lived. It is very short, and also there is no DNA replication here. No DNA replication. DNA replication is completely absent here. DNA replication is completely absent here. Understand now. Meiosis 1 is also called as reduction division. Why? Because in the meiosis 1 itself, 2n gets converted to n. Meiosis 1. While your meiosis 2 students, meiosis 2 is called as equational division. It is called as equational division. Why? Because your meiosis 2 is just like mitosis. Meiosis 2 is just like your mitosis. Done? Done students? Done. Now, I'll give you a small recap again. First we have the meiosis interphase G1 S G2, meiosis 1 interkinesis, meiosis 2, prophase, metaphase, anaphase, telophase, no DNA replication, short-lived, prophase 2 and metaphase 2, anaphase 2, telophase 2. Meiosis 2 is called as equational division. Why? Because it is just like mitosis. Now students, understand. This prophase 1 is right here, right? This prophase 1 is again further divided. This prophase 1 is again further divided into many categories. This prophase 1 is again divided into leptotene, zygotene, pachytene, diplotin and diakinesis. Yes sir. So Shreyas sir, look at that. Even on the Diwali, Shreyas sir is here. All the students are also here and studying really well. Okay. Understand now students. This prophase 1 is again further divided. This prophase 1 is again further divided into leptotene, zygotene, pachytene, diplotin and diakinesis. See this. So here we have meiosis 1, meiosis 2, prophase 1, metaphase 1, anaphase 1, telophase 1, meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. This prophase 1 is again further divided. It is further divided into leptotene, zygotene, pachytene, diplotin and diakinesis as well as diakinesis. Yes or no? Tell me students. There's a small trick here to remember this. That is, let zebra play during the day. L for leptotene, zygotene, pachytene, diplotin and diakinesis. That is called as your prophase 1. Now students, quickly, Raise your hands. How many of you find prophase 1 difficult to understand? And I will make it easy for you today. Raise your hands, all of you. Raise your hands. True, sir. Raise your hands, all of you. Raise your hands. How many of you feel the entire process of your prophase 1 is difficult to understand? NEET teen. Raise your hands, all of you. Raise your hands. How many of you feel it is difficult to understand the prophase 1? Especially prophase 1. Tell me. How many of you feel it is difficult? Small difficulty is there. Even if small difficulty is there, I will make it very easy. Just follow through, listen to my words, and it becomes very easy to understand. Can we start now, students? Can we start? Yes or no? Can we start now, students? There is a remote. Okay. Now, I hope the basic idea is clear to all of you. Meiosis, interphase, meiosis. Meiosis 1, prophase 1. Right now, we will start the prophase 1 leptotene. Prophase 1 leptotene first. Leptotene. Okay. So, what exactly happens in the case of leptotene? In the case of leptotene, chromatid condensation and chromosomes are visible under light microscope. That's all. That is, first stage in the case of prophase, chromosomes gradually become visible under the light microscope. Under the light microscope. Under the light microscope. Under light microscope, it becomes very visible. First point done, all of you. Tell me, first point done. Yes or no? Compaction of chromosomes continues. Then followed by zygotene. That's all. That's all is you are going to be your leptotene done. Leptotene done. Now, what exactly happens in the case of your zygotene? Understand. Okay. Now, students, all of you know. Tell me, a gene has how many alleles? From the previous chapter, we learned that a gene has 2 alleles. One allele from the mother, one allele from the father. 2 alleles. Similarly, in our body, we have how many chromosomes? We have 46 chromosomes, yes or no? 46 chromosomes are there. Okay. We have 46 chromosomes. That is 23 pair. 23 pair of chromosomes. One set coming from mother, one set coming from father. So, we have totally 23 pair. We have 23 pair of chromosomes. Pair of chromosomes. Now, students, what happens is we will do match up. We will do match up now. So, what exactly happens in the case of zygotene? In the case of zygotene, one chromosome, one more chromosome, they will start pairing up. They will start pairing up. And the process of pairing up, is called as synapsis. It is called as what? Synapsis. For example, students, here I have one chromosome. Let me change the color. Here I have one chromosome. Okay. Here I have one chromosome. Here I have one more chromosome. Okay. Now I have 2 different chromosomes here. Let me change the color, students. It's not very widely visible. Make it green. Okay. So these are called as what? They are called as homologous chromosome. I will call them homologous chromosome. Homologous chromosome. So what exactly is happening here? First point is what? Pairing of homologous chromosomes. 2 chromosomes are pairing up, and the process of pairing up is called as synapsis. That is, chromosomes start pairing together, it is called as synapsis. First point done. Now students, understand, when the chromosomes are pairing, between the 2 chromosomes, there is a complex formation. There is a complex formation between the chromosome, and I will call this complex. I will call this complex as synaptonemal complex. Synaptonemal complex. Synaptonemal complex is taking place between the two chromosomes. Done. First is called as synapsis. What is synapsis? Two chromosomes coming together is called as synapsis. Between the two chromosomes we have synaptonemal complex. Now students, this stage right here is called as bivalent stage. It is called as bivalent stage, or also called as tetrad stage. It is also called as what? Tetrad stage. But why? Why do we call it bivalent, or why do we call it tetrad stage? Why? Answer is very simple. We call this as bivalent. Why? Because of two chromosomes. Because of two chromosomes we call it bivalent. 1, 2 chromosomes. Why do we call it tetrad stage? We call it tetrad stage because 4 chromatids. 1, 2, 3 and 4. 4 chromatids called as tetrad stage. Then so all of you know now. We will call this stage as zygotine. 3 main points. First one is synapsis, 2 chromosomes coming together. Second point is going to be synaptonemal complex formation. Third point is called as what? Bivalent or tetrad stage. Now students, very important concept. This bivalent or tetrad stage, formation is happening in zygotine. But it is clearly visible in the next stage. This bivalent or tetrad is clearly visible. This bivalent, this is called as synaptonemal complex formation. It is clearly visible in the Pachytene stage. Bivalent chromosomes clearly appear as tetrad. Where? In the case of Pachytene stage. In the case of Pachytene, they become much more clearly visible at Pachytene stage. Done. Longer than previous one. Now what exactly happens here, students? Understand, there is something called as crossing over is happening here. Let me draw and show you. Very easy to understand. Here I have one chromosome. Okay. Here I have one chromosome. Second chromosome is right here. 2 homologous chromosomes are packed together, and between the chromosomes there is a synaptonemal complex. Okay. Now what exactly happens, friends? These two are called as what? One chromatid from here, they are called as non-sister chromatid. I will call them non-sister chromatid. Okay. Now what is happening here? Understand. Okay, till there is still clear. Okay, now they are crossing over. Okay, now next what is happening? In the next step is very crucial to understand. So what happened here, students? Are you able to see, all of you? All of you able to see in the diagram, in the this thing? Here we had 2 homologous chromosomes coming together, synapsis. Now here you can see their chromatids are crossing over. Now can you see there is something called as exchange of genetic material. There is something called as exchange of genetic material between non-sister chromatids, non-sister chromatids of homologous chromosomes, of homologous chromosomes. This entire process is called as what? It is called as crossing over. Crossing over. Definition is right here. Definition is right here. That is called as crossing over. So crossing over happens where? Crossing over happens in the case of packatine. In the case of packatine. Okay. Now students, understand, in this entire structure there is something called as, at the site of crossing over. At the site of crossing over here, there is some form of nodule formation. At the site of crossing over, there is some form of nodule formation. We call that recombination nodules. We call that recombination nodule formation. Recombination nodules. We call that recombination nodules. Recombination nodules. And students, understand, this entire process is enzyme mediated. Enzymes are involved, and the enzyme involved here is, can anyone tell me? Starts from R. Recombinase. Recombinase is the enzyme which is involved here. Recombinase is the enzyme which is involved here. Recombinase enzyme. So what we learn here, students? We learn that the first point is what? First point is going to be bivalent chromosome becomes very visible. Second main point is crossing over. Crossing over. Okay. Third main point is recombination nodules. And the fourth point is enzyme, that is going to be recombinase enzyme. Recombinase enzyme. This entire process is called as, happens in the case of your packatine. In the case of packatine. Done. Tell me, students, do all of you understand packatine? Yes or no? Tell me. You know, all of you understand packatine? Crossing over. Crossing over. Exchange of genetic material between non-sister chromatids. Between non-sister chromatids. Crossing over. Can you see? Homologous chromosomes. Crisscross. Exchange of genetic material. Exchange of genetic material. This is what is happening. Crossing over is happening at packatine stage. Packatine stage. Here. Packatine stage. Done. Now what exactly happens in the case of diplotin? Diplotin. The first point you understand, students. First point you understand. First we will draw the chromosomes and I will show you. Students, tell me, in between the chromosomes, what do you call this structure here? What is this? That is called as synaptonemal complex. That is called as your synaptonemal complex, right? Synaptonemal complex. Now, what exactly happens in the case of your, diplotin? In the case of diplotin is that this entire synaptonemal complex will completely disappear and the chromosomes will separate out. Entire chromosomes will separate out. You see here, the entire synaptonemal complex is completely disappearing and the entire chromosomes are separating. Now, but they are not crossing completely. They are not separating completely. At point of crossing over, this is called as point of crossing over. Point of crossing over. At the point of crossing over, can you see they are still joined, and here we find a X-shape mark. We find a X-shape mark right here. This X-shape mark is called as chiasmata. We call this X-shape mark as chiasmata. Chiasmata. Here. The X-shape mark here is called as chiasmata here. The X-shape mark called as chiasmata. apart from chiasmata, the entire chromosomes are completely separating out. Is that point clear, students? Is that point clear? X-shaped mark is called as what? Chiasmata. Everywhere else it is separating out. Everywhere else it is completely separating out. Okay, done. Now, next is called as diplotin. What exactly happens in the case of diplotin? In the case of diplotin, the X-shaped mark also starts to terminate. The X-shaped mark is also called as terminating. That is called as termination of chiasmata. The X-shaped mark also is separating out, and we have two separate chromosomes. That is final stage of prophase 1, termination of chiasmata. Chromosome contains fully, mitotic spindle assembles, nucleolus disappears, membrane breaks down and represents the transition to the metaphase, and that is the end of prophase 1. So what we learned right now, diplotin, leptotin, zygotin, pacotin, diplotin, diakinesis, all the 5 stages were under your prophase 1. Done and dusted. Now we will go to metaphase. Very simple. Now students, metaphase is a little easy to understand, but I'll give you a perspective. So in the case of mitosis, and in the case of meiosis, the difference you understand. Okay. This is mitosis. This is in the case of mitosis. This is in the case of meiosis. In the case of mitosis, all the chromosomes were at the center. All the chromosomes aligned at the center. Okay. All the chromosomes were aligned in the center, equatorial plate in mitosis. But in the case of meiosis, students, do we have homologous chromosomes right now? Yes or no, tell me. Right now, in the case of meiosis, we have 2 chromosomes. At the end of diplotin, did we have 2 chromosomes? Yes or no, tell me. At the end of diplotin, did we have 2 chromosomes like this? At the end of diplotin stage, we have 2 separate chromosomes now. Yes. So these 2 chromosomes need to be aligned on the equatorial plate now. Okay. Let me align them. For example, the 2 chromosomes are like this. Now I will take them and I will align them on the plate. Now tell me, students, if I have to align 2 chromosomes on the plate like this, will I need only 1 equatorial plate or will I need 2 equatorial plates? Tell me now in the chat. If I have to take them and align them here like this, will I not require 2 separate plates now? So here we have 23 chromosomes, here we have 23 chromosomes. Understand? Okay, totally 46 chromosomes. All the 46 chromosomes are arranged like this, 23 on top, 23 on the bottom. Homologous paired are completely arranged in the plate. Okay. Now in the case of mitosis, here we have the centrioles, pericentriolar amorphous substance, and we had spindle fibers. Spindle fibers attaching. Now students, if you notice here, every chromosome has 2 spindle fibers. But in the case of meiosis, it is a little different. In the case of meiosis, what happens, you check. In the case of meiosis, 1 chromosome will have only 1 spindle fiber. It will only have 1 spindle fiber. Now students, you tell me, all of you, imagine. I'll give you all of you imagination power now. Imagine. If I pull the spindle fiber, if I hold the spindle fibers, if I pull from here, will the centromere split? Tell me, students. Will the centromere split here? Hold the spindle fibers, pull it apart, centromere split, and chromosomes will be separate. Okay. Now imagine here. if I hold a spindle fiber and if I hold a spindle fiber and if I pull them apart, will the centromere split now? Tell me, will the centromere split? No. Simply what will happen? Simply these two chromosomes will be pulled down, these two chromosomes will be pulled up. Yes or no? Tell me students, yes or no? If I pull them from this side, the two chromosomes will be pulled. The centromere will not split. If I pull them from this side, the two chromosomes will move up, but the centromere will not split. The centromere will not split at all. Centromere will not split. So that is what is happening in the case of your anaphase. In the case of anaphase, what is happening? In the case of anaphase, homologous chromosomes separate. Sister chromatids remain intact or associated. They will remain intact. Only and only homologous chromosomes are separating. Can you see here? This is one entire homologous chromosome. This is one entire homologous chromosomes. Homologous chromosomes are being pulled apart. Okay. Now students, that is your anaphase. That was your anaphase. Then we have your telophase. Nucleolus, nuclear membrane reappears. Cytokinesis occur, and we get what? We get dyad of cells. Now what is the meaning of that? This is a dyad of cell. This right here is called as your dyad of cell. Dyad of cell. Okay. So we have one cell, 2n. After meiosis, we get two cells called as dyad of cells, and both are going to be haploid in nature after meiosis 1. After meiosis 1, we have dyad of cells. Can you see here? In the case of anaphase, what happened? Here we have 23 chromosomes and 23 chromosomes on top. 23 chromosomes on top. So 23 went this side, 23 chromosomes came this side. In the plate, can you see here? 23 chromosomes here, 23 is here. So 23 going on top, 23 coming on bottom. Eventually, what is happening? Eventually, the two cells is dividing. Can you see? 23 on top, 23 on the bottom. Nuclear division, cytokinesis, and we get two cells. We get two cells, that is 23 and 23, N and N. That is the end of meiosis 1. Did all of you understand the concept of meiosis 1 instead of just by hearting it? Tell me yes or no. Students, did you understand? Did you understand? Tell me. Beautiful, right? Beautiful. This is called as meiosis 1. Meiosis 1. Meiosis 2 is very easy. Meiosis 2 is very easy. Why? I'll come back to this later on, students. I'll come back to this later on. Then we have meiosis 1 is done. Then we have interkinesis, no DNA replication, followed by prophase 2. Okay. Prophase is just like your prophase. Your meiosis 2 is just like mitosis. So what will happen in prophase 2? Condensation, chromosome becomes compact, that's all. Metaphase 2, what happens? Chromosome align at the equator. Chromosomes align at the equator. Microtubules will attach the kinetochore. Done. What happens in the case of anaphase 2? In the case of anaphase 2, just like in the case of mitosis, centromere split, two separate chromosomes, right? So in the case of anaphase, splitting, and we are getting two different chromatids, we are getting two separate chromatids, and again each chromatid will act like chromosome, here, done, Then we have telophase 2 again, nuclear membrane reappears, nucleolus appears, cytokinesis occur, and finally we get 4 cells. Okay. So what exactly happened here, students? We had a 2n, it gave birth to 2 cell, n and n. This n also divided 2 cells, n. And this right here First one is meiosis 1. Here we have meiosis 2. Meiosis 2. Meiosis 2 is just like mitosis. Just like mitosis. Okay. Daughter cells. Okay. Prophase 2, metaphase 2, anaphase 2, telophase 2. This is the entire DNA content. This is the entire DNA content. Students, I'll give you a recap of this. Listen to me very carefully. Listen to me very carefully, students. I'll give you a small recap here. Okay, small recap I'll give you. First we had G1. G1, 46 chromosomes, 46 chromatids, 40 DNA content. 40 DNA content. Okay, 40 DNA content. Now, then we have S46. Chromatids will become double, right? From one chromatid to become double chromatid. So 92. DNA content is also doubling. 2N2C to 2N4C. So 80. In G1, prophase, metaphase, anaphase, it will remain the same. It will remain the same itself. There is no change much. But as soon as we come to telophase, what exactly happens in the case of telophase, students? In the case of telophase, what happened? 46 chromosomes, 92 chromatids, did it divide into two separate categories? See here. In the case of telophase 1, what happened? When we came to telophase, this entire thing has how many chromosomes? This entire thing has 46 chromosomes. Entire thing had 46 chromosomes, right? Till here it was 2 and 4C. Till here it was 2 and 4C, right? Till here it was 2 and 4C. Now if I am adding out of 46 chromosomes, I am sending 23 here and 23 on top. What will happen per nuclei? Tell me. Per nuclei, this is going to be now N. This also is going to be N. 4C can divide into 2C and 2C. So 2 and 2C is dividing into N2C, N2C. So out of 46 chromosomes at the telophase, 23 is coming on bottom, 23 is going on top. So N2C, we get N2C. So what do we get? We get N2C. And this is the point where we have meiosis 1, and in the case of meiosis 1, from 2N we are making N. Same thing is continuing in your prophase, that is metaphase 1 everywhere, 23, 46, 40. But as soon as we come to anaphase 2, and remember students, anaphase 2 is just like your anaphase of mitosis. Every single chromatid will act like a chromosome now. Can you see here? So in anaphase 2, 46 chromosomes and 46 chromatids. Why 46 chromosomes here? Because every single chromatid is acting like a chromosome. So all the chromatids will convert into chromosomes now. That's why 46 here. DNA content will remain the same. It will remain the same. Then we come to telophase now. Entire cell wise, telophase will be how much? Entire telophase is 46 chromosomes. 46 chromosomes with a single chromatid. Okay, single chromatid. Now there are 46 chromosomes here. 46 chromosomes with a single chromatid. Now half is going on this side and half is going on this side. What do we get, students? Now tell me. Now what is the DNA content here? DNA content is how much? DNA content is 40. DNA content is 40. That is NC. So right now we are what? Right now we are at 2 and 2C. 2 and 2C. This entire thing is 2 and 2C. Entire thing. So can I tell if 2 and 2C is dividing into 2? Dividing into 2, 46, N is going here, N. 1 N is coming down, N. 1 C is going on top, and 1 C is coming on the bottom, C. So we get NC. Now we end up getting NC. 2C is dividing into CNC. CNC. Okay. So we end up getting NC. NC. Okay. So students, go through this one more time. Any doubt anywhere you can ask me always. You can always ask me. Okay. This was 2 and 2C. 2 and 2C divided into NC. NC, NC. 2 of them. Okay. 2 of them. So that is your meiosis. So did you notice in the case of meiosis, there was a change in chromosome number. So can I call it reduction division? Reduction division. Yes, I can call it reduction division. I can call it reduction division. Reduction division. Now students, if you notice here, one cell is at 40 DNA content. 40 DNA content. Okay. One cell is 40 DNA content. Okay. 40. One cell. Now it is making 4 cells. Each one is how much? 20. 20, 20, 20, 20. How does it make sense then? How does it make sense? From one cell that is 40, how are we getting 4 cells which are 20, 20, 20, 20? What is the answer to this question? What is the answer now? Tell me, students. Answer is very simple. Answer is simple. From G1 to S, the 40 became 80. The 40 got converted to 80. Now tell me, students, can the 80 be divided into? Can the 80 be divided into 20, 20, 20, 20 now? 20, 20, 20 and 20. 40, 40, 80. Makes sense now. Everything makes sense now in biology. So this is the beauty of biology. This is the beauty of biology. When you follow things, you understand. No need to by heart any of this. Okay. So that is the beauty of your cell division. Significance is very simple. Significance is very simple again. First one is gamete formation during sexual reproduction. Okay. Genetic variability. Okay. That will lead to variation. That will lead to variation and evolution. And evolution. Okay. And evolution. Now solve this question, students. Let's see how many of you can solve this question. I'm going to move outside. I'm going to move out of the frame and let you read the question. Let you read the question now. Solve the question, students. Quickly solve the question now. See, all of you know. Tell me, recombination nodules, packaging. Yes or no? Tell me. Recombination nodules or recombinase enzyme or recombination or crossing over, everything is happening at packaging. So B is going to be 4. B is going to be 4. So B is going to be 4. How many options are there? Tell me, students. Only one option. Only one option. Option number 2. Option number 2. Yes or no? Yes. A is going to be 2. A is going to be 2. Complete termination of chiasmatic diakinesis. Zygotine, what is happening? Zygotine is synaptonemal complex formation. Yes. Leptotene is what? Chromosome look like thin thread. First stage. First stage. Next question. Chromosome become gradually visible under light microscope. Leptotene. Yes. The beginning of diploidine stage is recognized by dissolution of synaptonemal complex. Yes or no? In diploidine we have chiasmatic formation and dissolution of synaptonemal complex formation. That is diploidine. Correct? Both are correct. Both are correct. Next question. Chromosomes start pairing together to form synaptonemal complex formation. We just now learned A is going to be 2. A is going to be 2. Chromosome become visible in light microscope. That is leptotene. B is going to be 4. A is going to be 2. B is going to be 4. Okay. 4 chromatids of bivalent chromosome becomes distinct and recombination nodules. See, recombination nodules directly packaging. Directly packaging. C is 1. C is 1. Diploidine. Dissolution of synaptonemal complex. We learned here. Dissolution of synaptonemal complex is a diploidine. Diploidine. So diploidine. So A is going to be 2, B is going to be 4, C is going to be 1 and D is going to be 3. Option number 4. Option number 4. Next question. The process of appearance of recombination nodules. Students, recombination nodule answer should be what? Packaging. packaging. Direct question, direct question. See, every single question is very direct. If you practice enough, every question is very easy, free marks. Every single question is laddu question. Which of the following stage of meiosis involves division of centromere? Division of centromere. Tell me, centromere splitting in the anaphase 2. Anaphase 2. Is it dividing in anaphase 1? Tell me yes or no. In anaphase 1, is it dividing? No. In anaphase 1, there is separation of homologous chromosomes. No division of centromere. Next question. Select the correct statement. Tetrad formation seen during leptotin? No. During anaphase, I'll move out of screen so you can read very carefully. During anaphase, centromere split and chromatids separate? Yes. Termination takes place during packaging? No. Nucleolus Golgi complex ER are reformed in telophase? Yes. Crossing over takes place between sister chromatids of homologous chromosomes? No, non sister chromatids. Tell me the answer now. B and D? B and D? Yes or no? B and D? Yes. No. Same question again, repeat. Dissolution of synaptoterminal complex, diplotin. Same question, same question. During which stage of mitosis and meiosis, respectively, centromere split? Centromere split. Tell me, students. See, centromere splitting in the case of your anaphase, all of you know, anaphase. And also in the case of anaphase 2, because it is just like mitosis, also in anaphase 2. Next question. Select the substages of prophase 1. Leptotin, zygotin, packaging, diplotin, diakinesis. That is the process. Leptotin, zygotin, leptotin, zygotin, packaging, diplotin, diakinesis. Clear. Which of the following never occurs during mitosis? Never occurs in mitosis. Coiling and condensation of chromatid occurs. Spindle fiber attached to kinetochore happens. Movement of centriole to opposite poles happens. Pairing of homologous chromosomes does not happen in mitosis. Does not happen in the case of mitosis. Why? Because in the case of mitosis, there is no homologous chromosomes at all. That is happening in the case of meiosis. Appearance of recombination nodules on homologous chromosomes in meiosis happens in packaging during crossing over, site of crossing over. Regarding meiosis, which statement is incorrect? Okay. Four haploid cells are formed at the end of meiosis 2. There are two stages in meiosis, meiosis 1, meiosis 2, yes. DNA replication occurs in S phase of meiosis 2, students. Does it happen in the case of S phase of meiosis 2? Tell me. How many questions are there, students? How many questions are there still? Many questions are there. Don't worry. Does it happen in the case of interkinesis? Does it happen in interkinesis? No. In which, in prophase 1 of meiosis, chromosomes start pairing together, synapsis takes place. Synapsis, which stage? Synapsis, synaptoterminal complex formation happens in the case of your zygotin. The beginning of diplotin is recognized by dissolution of synaptoterminal complex, yes. X formed chiasmata, yes. In oocyte of some vertebrates, diplotin can last for many months or years, yes it can. But it is explaining, it is not explaining. This point is very important to understand. Remember this point. Diplotin can last for many years. When I am teaching you about oogenesis, the primary oocyte is arrested at diplotin stage, right, for many years. Next question. The oocyte of some vertebrates got arrested in many years or months in diplotin. Okay, let's check one by one, quickly. Students, time starts now, go. Okay, mistake is the answer. I sat down and the answer came off. Answer is on the board, students. Very easy question again. Very easy question. Let's go next question. In meiosis crossing over, exchange genetic material between the homologous chromosomes is enzyme. Which enzyme is involved? Which enzyme is involved? It's an RNA enzyme. Recombinase enzyme. Recombinase enzyme. Not RNA, it's recombinase. Which stage of mitotic prophase shows termination of chiasmata? Termination of chiasmata happens in the last stage. Diakinesis. Diakinesis. After meiosis 1, the resultant nodal cells have. After meiosis 1, students. After meiosis 1, same amount of DNA as in parent in the S, twice the amount of DNA in comparison to haploid. I'll give you a small clue. It is N2C. Same amount of DNA in comparison to haploid gamete. Haploid gamete has how much? NC. Not same. 4 times the DNA content? No. Tell me, students, tell me the answer to this question now. Very good question. First we have 2 and 2C. From 2 and 2C, what do we get? From 2 and 2C, we become NC. N2C. N2C. And finally, NC. This is after meiosis 1. After meiosis 1. This is after meiosis 2. Twice the amount of DNA NC, twice the amount of DNA in comparison to haploid, right? In comparison to haploid, twice the amount of DNA. Here we have N, we have only C here. Here we have 2C. Okay. Is it same amount? No, it's not same amount. 4 times, it is not 4 times. Same amount in the case of compared to S. Same amount of DNA in compared in parent S. No. S will be how much? S will be how much, students? S will be 2 and 4C. Is it same amount here? Not the same amount. Not the same amount. So answer is number 2. Okay. I think we will stop here, students. The questions are very same questions are repeating again everywhere. Everywhere we have same number of questions. And I think we'll stop there, students. Questions are very easy again. You will be able to solve every single question. The PDF will be available on Telegram channel. Download the PDF, see the questions, write the questions, solve them. If you are not able to solve any questions, come back to me, ask me, and as I promised, in a compact session, we finish the entire cell cycle, entire chapter of cell cycle and cell division in just almost 2 hours. 11, 12, 12 to 1, almost 2 hours, 10 minutes, 15 minutes, we finish the entire chapter. So that was the beauty of Tejas series, where we can finish everything in very short duration. Students, the sale is going on still. Go check it out. Look at the sale. You will be able to get books on discount on books, discount on flashcards, discount on the DNA batch for free in many cases. So go check out the links. Link in the description. Let me know in the comment section how was today's class. Did you enjoy today's class? If you did, do not forget to fill up, let me know in the comment section. When you add the comments, it makes me happy, and all of you, it gives us motivation. So thank you so much, students. Smash the like button, share the video to your friends who are still confusion in the cell chapter, and I will see you in the next class. Thank you so much. Bye bye. Take care, students. And also, next chapter is going to be biomolecules, compact session of biomolecules. So I will see you there. Bye bye. Take care, all of you. Have a great day ahead. Bye bye.